Assalamualaikum and hello to everyone. So today I would like to introduce our team members. So firstly, my name is Farah Emilia binti Azmi, and this and we are from AT222
Now we move to the introduction. Modern technologies have been widely used in the agriculture sector, which to improve the crop yield. This modern breeding may use techniques of molecular biology to select and insert desirable traits into plant. Modern breeding is a genotype-based method of breeding. It is done by isolating a gene from an organism that has the features that we want to impart into a plant. Through this technique, the inserted genes into crops able to make the crop resistant but harmless to humans.
In plant breeding, there are two types of breeding, namely modern and conventional breeding. Both have their own different and their own characteristic. Firstly is conventional breeding. Conventional breeding is the process of developing new plant cultivar without the use of futuristic molecular breeding technologies. Conventional breeding is also a technique where plants are selected based on superior performance on selected character. And it also a technique that requires a long period of time to release a new cultivar. Among of the methods used in conventional breeding is hybridization. And in conventional breeding, identical pairing do not produce a variation as there will be no segregation of gametes. Next is that it is less effective because to do a good plant breeding it requires skill, art and subjective analysis of the breeder. For modern breeding, it uses technique of molecular biology to select. Compared to conventional breeding, modern breeding is more accurate to determine the pure gene line because in modern breeding, it uses a genotype-based method and modern breeding only require a short period of time to release a new cultivar. Apart from that, in modern breeding, it uses genetic selection and high throughput phenotyping or HTP method to produce a new cultivar. However, modern breeding requires more technical skill and genetic science knowledge compared to conventional breeding which only requires basic knowledge. Lastly, modern breeding is more effective and trusted because before releasing a new cultivar, the breeder has done a scientific research and scientific finding. Next is the method of modern breeding. Among the methods that are often used is genomic selection. The use of genomic selection can produce a lot of fruit because in this genomic selection, it can help in increasing grain yield in a shorter time. Among the objectives of this method is to make an assumption on performance of future offspring based on DNA. Next is how to obtain breeding value. There are two methods that can use, which is penalized regression method and Bayesian method. And the value can be calculated by using two datasets, which is training population and validation population. Next is that genomic selection can act as effective measure for improvement of quantitative trait. This can happen due to the increase in the price of phenotyping of crop. The following technique is a marker-assisted selection. Marker-assisted selection is a way of discovering the genomic areas responsible for the expression of desirable trait in plant by using multiple DNA markers. The availability of numerous DNA markers has also made it easier to incorporate marker-assisted selection into breeding. Aside from that, DNA can aid in the identification of allele variation. Next, marker-assisted selection can be used to choose quality like as grain yield, blossom colors, and seed characteristics. The marker-assisted selection method is also effective in selecting traits that are less heritable, expensive, and time-consuming. However, this method will be effective in the case of selecting a few quantitative traits that control a significant amount of variation in a population. And several DNA markers that are always used in this method are restriction, fragment length, polymorphism, and single nucleotide polymorphism.
The purpose of modern breeding is to develop a plant variety with specific properties for survival in their local region. Next is to create environmentally sustainable, higher yield and less expensive yield. Next is to develop a plant with more variants that are more enriched with healthy components than the wild type species. Next is to develop a species that help to limit post-harvest crop loss. Next is to breed new varieties of plant to increase biodiversity. There are two types of plant that develop from modern plant breeding. First is herbicide-tolerant soybean. It was introduced by Monsanto in 1996 by using genetic engineering. Herbicide will only kill the surrounding weeds but leave the soybean intact. Next is golden rice. Rice that accumulate beta-carotene in rice grain by using transgenic technology. Once inside a human body, beta-carotene in the golden rice will convert into vitamin A. Plant breeding programs are changing as science and technology advance. Traditional breeding methods are made faster and more effective by incorporating other disciplines of science, such as genetic and animal husbandry. The reduction in variety caused by selection of traits function as a bottleneck in plant breeding. Crop with high yield is chosen and grow in a broader area than the crops that were once culturally important.